All right, let's talk about the engine of life itself. Today, we are diving deep into the Krebs Cycle, which is basically the central power station that's humming away inside pretty much every single cell in your body. It's an absolutely incredible piece of molecular machinery. I mean, have you ever really stopped to think about it? How does that one bite of food you just ate get turned into the actual energy you need to, think, to move, to live? Well, the answer is hiding inside this beautiful, microscopic, and incredibly powerful engine. So here's how we're going to tackle it. First, we'll get straight to the point. What is this thing? Then we'll look at the basic design, the engine's blueprint. After that, we'll take a simplified tour of the cycle itself, and I promise to make it easy. Then we'll count up the actual energy it produces. And finally, we'll uncover its secret double life as a molecular factory. Okay, so first things first. What exactly is the Krebs Cycle? You've definitely heard the name before. In fact, you've probably heard a few of its names, which is kind of the first clue to just how important this process really is. So yeah, it's got a few aliases. Officially, you might see it as the citric acid cycle because, no surprise, citric acid is the very first molecule made in the chain. You'll also hear it called the TCA cycle, but honestly, most of us just know it as the Krebs cycle. At its heart, it's a series of 8 chemical steps that happen deep inside the cell's powerhouse, the mitochondrial matrix, and its whole job is to tear apart fuel molecules to release their stored energy. And a process this important wasn't figured out overnight. It was like a huge puzzle, pieced together back in the 1930s. You had brilliant minds like Albert Szent-Györgyi laying the groundwork, and then Hans Krebs came along and put the final pieces together, revealing the full, beautiful loop. And yeah, they both won Nobel Prizes for it. That's how fundamental this discovery was. All right, now let's pop the hood and see how this thing actually works. Now, the best way to understand the Krebs cycle and to actually remember it is to stop thinking of it as some flat, boring diagram in a textbook. Instead, I want you to picture it as a dynamic, whirring engine. Because at its core, any engine really only does 3 things, right? You put fuel in, you get usable energy out, and you get rid of the waste. The Krebs cycle is exactly the same. The fuel is a little 2-carbon molecule called acetyl-CoA. The energy it extracts isn't heat or motion, but high-energy electrons. And the waste? Well, that's just carbon dioxide, which you are literally breathing out as we speak. Now this is probably one of the most important slides to really get. There are 2 key players here. Think of acetyl-CoA, the 2-carbon molecule, as the gasoline. It gets completely burned up every single time the engine turns. But oxaloacetate, the 4-carbon molecule, that's part of the engine itself. It's like the piston. It grabs the fuel, does the work, and then at the very end, it's reset, regenerated, and ready for the next drop of fuel. One is consumed, the other is recycled. That's why it's a cycle. Okay, this is the part where most students' eyes glaze over. They see 8 steps, 8 enzymes, 8 molecule names, and they just try to brute force memorize it. We are not going to do that. To make this stick, we're going to group all 8 of those complex steps into 3 simple, logical phases. Let's walk through it. Phase 1 is fuel injection. Our 2-carbon fuel acetyl-CoA combines with the 4-carbon piston oxaloacetate. 2 plus 4 gives us a 6-carbon molecule called citrate. Easy. Phase 2 is the energy harvest. This is where the work gets done. The engine systematically breaks down that 6-carbon molecule, snapping off 2 carbons as CO2. And with each snap, we harvest those precious high-energy electrons. Phase 3 is regeneration. We're left with a 4-carbon molecule, and this phase is all about reshaping it back into its original form, oxaloacetate, so it's ready to grab the next bit of fuel. And if you just follow the carbons, it makes perfect sense. We take a 4-carbon molecule, add 2 carbons from the fuel, and we get a 6-carbon molecule. Then, during the energy harvest, we lose those 2 fuel carbons as CO2, which takes us right back down to a 4-carbon molecule. The rest of the cycle is just about restoring that molecule to its starting form. It's an absolutely perfect, self-sustaining loop. So, we've put the fuel in, we've run the engine through a full turn. What do we actually get out of it? What is the real power that this engine is generating for the cell? First up, with every single turn of the cycle, we get 3 molecules of the stuff called NAD. I want you to think of each NAD molecule as a fully charged, high-capacity rechargeable battery. Next, we also get 1 molecule of FADH2. This is just another type of charged battery. Maybe think of it as a different brand, like a Duracell versus an Energizer. It does the same job. It carries high-energy electrons. And finally, the cycle spits out 1 molecule of GTP. Now, GTP is basically identical to ATP, which you know is the direct, spendable energy currency of the cell. So while the other products are charged batteries that need to be cashed in later, this is like getting a little bit of cash paid directly to you on the spot. So this is so important to remember. The main job of the Krebs cycle isn't to make a ton of ATP directly. Its main job is to create all these NADH and FADH2 molecules. They're like little energy taxis. Their whole purpose is to pick up the high-energy electrons that were stripped from the fuel and drive them over to the final stage of cellular respiration, the electron transport chain. That's where the big energy payoff happens. Now, for a very long time, that's where the story ended. The Krebs cycle was just an energy production line. But it turns out, the Krebs cycle has a massive side hustle. It isn't just an engine for burning fuel, it is a central manufacturing hub for building new things. The technical term for this is an amphibolic pathway. You know, amphi, like an amphibian that lives a dual life on land and in water. This pathway has a dual life too. It has a catabolic life, breaking molecules down for energy, and an anabolic life, using those same molecules to build things up. This is what makes it so unbelievably central to everything the cell does. So what kinds of things does it build? I mean, pretty much everything. The cell can literally pause the cycle at any point and pull intermediates right off the assembly line. It can pull out citrate to make fatty acids and cholesterol. It can grab alpha-ketoglutarate or oxaloacetate to build new amino acids. And get this, it uses succinyl-CoA to make heme, the critical part of hemoglobin that carries oxygen in your blood. It is a true metabolic crossroads. And if you thought that was cool, the story gets even more amazing. The latest research is showing us that these molecules aren't just passive building blocks. Intermediates like succinate and fumarate can actually pile up and act as signaling molecules. They can literally influence which genes in your DNA get turned on or off. They don't just fuel the cell, they are talking to it, helping to decide its fate. So let's just end with this thought. This elegant, powerful, dual-purpose cycle is found in nearly every single aerobic organism on this planet, from a tiny bacterium to a giant sequoia tree. It evolved billions of years ago and has been kept almost unchanged ever since. So what does that tell you about its importance? It suggests that this isn't just one way to power a cell. This might just be one of the fundamental blueprints for life as we know it.